All right, in this video, we're going to talk about proteins. Okay, so this is the third group of biochemical families, the first two being the carbohydrates and the lipids. Okay, proteins are the third group. Now, proteins actually can be used for energy, but that's not their primary function in the body. First of all, a protein is held together by a special kind of bond called a peptide bond. Why is it special? Well, because in this case, what we're doing is we're bonding a nitrogen to a carbon instead of a carbon to a carbon. It gets a special name. Essentially, it's still a dehydration synthesis or condensation reaction that puts the amino acids together. By the way, the amino acids are the monomers that hold together or that are put together to create proteins, which is why proteins have an amino end, which is where the amino group is, and a carboxyl end, which is the other side of the amino acid. These are the workhorses of cells because they do so much. This is the jack of all trades of molecules. They communicate between cells. They can transmit information through communication. They can also serve as receptors of information. They're there for structure. They're there for movement. They can be there for storage, transport. Antibodies are there for defense. And by transport, by the way, I mean transport of molecules like oxygen in the blood. Hemoglobin transports oxygen in the blood. And then, of course, as enzymes. Now, we're not dealing with enzymes here. We'll deal with enzymes in Unit 3. But for now, just know that they can act as enzymes. So the monomer here is the amino acid, and there are 20 amino acids that exist in living things. There could theoretically be more than 20 amino acids, but our proteins only use 20 amino acids. So all proteins found in all living things use the same 20 amino acids. And you can distinguish the amino acid by their side chain or R group. And I'll show you that in just a second. The physical and chemical properties of the side chain are going to determine certain things about the amino acid, like if they're hydrophobic or hydrophilic. Remember, that's water-fearing versus water-loving. Some are positive, some are negative. Usually if they're hydrophobic or hydrophilic, if they're hydrophilic, they're polar. If they're hydrophobic, they're non-polar, usually. All right, so here are the amino acids. This is the set of 20. And this is a nice chart because it separates them for you, the nonpolar side chains, the polar side chains, and then the electrically charged side chains. Notice the part in purple is the same. So here's the amino group, right? That's the amino group on one side. That amino group is bonded to a central carbon that's attached to a hydrogen, and then you've got the carboxyl group on the end. Okay, so that's the basic structure of the amino acid. And then the side chain here for the nonpolar side chains are in yellow. So this one, glycine, just has an H. Alanine has a CH3, and so on. And then here on these hydrophilic side chains, they're in green. And then the acidic negatively charged side chains are pink. The basic or positively charged side chains here are in blue. But those are the side chains. Sometimes you'll see an amino acid written with this purple part and then just the letter R here. And the reason why R is chosen is to show that there's a side chain there, but R is not on the periodic table anywhere by itself. And so it can't be confused with any other atom that you would find on the periodic table. All right, now this is where it gets a little bit more complicated. This is the AP specific stuff. And by the way, you don't have to memorize any of those side chains. Okay, that's not part of your curriculum. You may remember from Baby Bio that we talked a lot about how shape of a molecule, particularly with proteins, determines its functionality. There is a lock and key fit between proteins and the molecules that they interact with, whether that's as an enzyme or a receptor or a transmitter of information or whatever. Shape is super important where proteins are concerned because it's going to determine the functionality of the protein. there are 4 different levels that we can look at in terms of how that protein is put together. And the first level is the primary structure. And this is where we kind of stopped in baby bio. If you took the 9th grade honors course, we might have gotten a little bit into tertiary structure. But this is where we're talking about the sequence of amino acids. And we're reading this from amino terminus to carboxyl terminus. Okay, that's the order that you're reading them in line. That's the primary structure of the amino acid, of the protein. And that's what we're looking at here. So this is primary structure. Okay, if we were reading this, glycine, proline, threonine, glycine, et cetera, et cetera, et cetera, that's the primary structure all the way down to the carboxyl end. Okay, that's primary structure. And you can see the ball and stick model of this here as well. So here's the amino end, here's the carboxyl terminus here. Okay, now secondary structure is the coiling or folding that you get on the backbone of this molecule. Okay, now remember, the backbone of the molecule, the molecules are bonded together. Amino end of one is going to bond to the carboxyl end of another, right? So the side chains stick out from the main protein. You've got a chain with side chains sticking out. That main chain is composed of these purple parts of the amino acids all stuck together, which means you have this regular bonding pattern in the backbone of the molecule. And so you're either going to get a regular helix, like a twist, a coil, or a regular fold, like an accordion pleat on the backbone. Okay, that's the secondary structure. And the secondary structure is due entirely to backbone interactions. So here's what a pleated sheet would look like, and here's what a helix would look like. Okay, so there's those. All right, the next structure that's important is the tertiary structure. And I think what's hardest for students to understand is what's the difference between secondary and tertiary. Remember that the secondary is solely due to the backbone of the molecule. Tertiary structure gets very irregular because now what you're doing is you're taking into consideration the side chains that are sticking up and how they're going to interact with one another. This is best described as a contortion. It's this very irregular pattern. That's the other thing. Secondary structure is usually pretty regular. Tertiary structure is not. Okay, and this is due to attractive forces, intermolecular forces between the side chains. That could be van der Waals forces, hydrogen bonding, and so on. The fact that some things are hydrophilic versus some things are hydrophobic, it's going to cause that protein to kind of twist and contort into its unique shape. Okay, so these are irregular. You also get patterns that are unique to each protein. You have some proteins that have sulfur in their side chain, and they will create disulfide bridges as well. Okay, so let's look at tertiary structure. Okay, and this really is actually a summary of all the types. So here's the primary. The secondary structure is based on and tertiary is based on the regular and irregular folding patterns. Okay, so here's tertiary really. You see this particular normal hemoglobin beta subunit looks like this. This is the sickle cell beta subunit, what it looks like. Okay, but it's this weird contorted shape that you're seeing. That's really the tertiary structure of the protein. And then the quaternary structure of the protein is the overall protein that results from the fact that many proteins are actually not a single chain of amino acids, but are actually multiple. And that's what you were seeing in that slide. Hemoglobin actually has 4 polypeptide chains held together. And by the way, that term polypeptide refers to protein every time you hear it, because remember that peptide bond holds together the proteins, right? So a polypeptide is a chain of molecules held together by peptide bonds. So you have 4 polypeptide chains, and they're held together with something called a heme group that has iron in it. Now, notice a lot of times the interactions that you see here, particularly in the secondary and tertiary structure, are attractive forces, intermolecular forces. It's very, very easy to mess with those with high temperature, extreme salinity, and pH changes, right? And when that happens, you can denature the protein. And that means that one of these three factors has interfered with those intermolecular forces that are holding the protein into its shape, and so the protein becomes misshapen. Okay, so let's go back to this diagram. Here's the quaternary structure. Hemoglobin has 2 beta subunits and 2 alpha subunits, and that's what you're seeing there. Okay, and when hemoglobin is normal, it looks like this, and it's inside of a nice, plump, fluffy red blood cell. And when hemoglobin is misshapen, when the beta subunit of hemoglobin is misshapen, then what you get are hemoglobin molecules that stick together and they form this, this sickled molecule that you can kind of see behind my picture.